Welcome to this scientific lecture on lab-on-a-chip technology for biosensing applications. Today we're going to explore one of the most revolutionary technologies in modern diagnostics and biomedical research. So what exactly is a lab-on-a-chip? Let me start with a simple definition. Lab-on-a-chip, often abbreviated as LOC, is a miniaturized device that integrates multiple laboratory functions on a single microchip, typically ranging from a few millimeters to a few centimeters in size. Think of it this way. Imagine taking an entire laboratory with all its equipment, reagents, and analytical instruments, and shrinking it down to fit on a chip the size of a credit card. That's essentially what lab-on-a-chip technology accomplishes. Here you can see the concept visually. A traditional laboratory with multiple separate instruments gets integrated into a single compact chip. Now let's break down the key components that make up a typical lab-on-a-chip device. First, we have microfluidic channels. These are networks of tiny channels, often just tens to hundreds of micrometers wide, that transport fluids through the device. Second, microvalves and micropumps control the flow and mixing of fluids, much like the valves and pumps in a traditional laboratory setup, but at a microscale. Third, detection units that can be optical, electrochemical, or magnetic sensors, depending on the application. These are the eyes of the chip that detect the analytes of interest. Fourth, sample preparation zones, where biological samples are filtered, concentrated, or separated before analysis. Here's a simplified schematic showing these components. You can see the microfluidic channel running through the center, with sample particles flowing through it, valves controlling the flow, and sensors detecting the analytes. Now you might be wondering, how does fluid behave differently at this microscale? Let's talk about the physics of microfluidics. At the microscale, surface forces dominate over inertial forces. This is fundamentally different from what we experience in our everyday macroscopic world. The Reynolds number, which characterizes flow regimes, is typically very low in microfluidic systems, usually less than 100. This means the flow is always laminar, never turbulent. The Reynolds number is defined as rho times versus times L divided by mu, where rho is fluid density, v is velocity, L is characteristic length, and mu is dynamic viscosity. Laminar flow enables extremely precise control over fluid behavior and creates predictable mixing patterns. This is crucial for reproducible biosensing. Now let's explore the exciting biosensing applications. What can lab-on-a-chip technology actually detect? First, disease biomarkers such as specific proteins, nucleic acids like DNA or RNA, and metabolites that indicate the presence of diseases like cancer, diabetes, or infectious diseases. Second, pathogens including bacteria, viruses, and parasites. This is especially important for point-of-care diagnostics in resource-limited settings. Third, circulating tumor cells. These are extremely rare cells shed from tumors into the bloodstream, and lab-on-a-chip devices can isolate and analyze them for cancer monitoring. Fourth, drug screening and toxicity testing. We can test how cells respond to various drug candidates in a high-throughput manner. Let me give you a concrete real-world example that many people are familiar with: glucose monitoring for diabetes management. Traditional glucose testing requires a significant blood sample, hours of laboratory processing, expensive equipment, and trained personnel. In contrast, lab-on-a-chip glucose monitors need only a tiny drop of blood, provide results in seconds, are portable, and can be used by patients themselves at home. Now let's discuss the various detection methods employed in lab-on-a-chip biosensing. How do these devices actually sense the presence of target molecules? Let's start with optical detection methods, which are among the most commonly used. Fluorescence detection involves labeling target molecules with fluorescent dyes. When excited by light of a specific wavelength, these dyes emit light at a different wavelength that can be detected. Absorbance measurements detect how much light of a specific wavelength is absorbed by the sample, which correlates with the concentration of the analyte. Surface plasmon resonance is a label-free technique that detects binding events between molecules on a metal surface by measuring changes in the refractive index. Here's a simplified illustration of fluorescence detection. Excitation light from above causes the fluorescent molecules in the detection zone to emit light, which is measured by a detector below. Now let's look at electrochemical detection methods, which are particularly useful for point-of-care devices. Amperometry measures the current produced by oxidation or reduction reactions at an electrode surface. The magnitude of the current is proportional to the analyte concentration. Potentiometry measures voltage changes that occur when target molecules interact with ion-selective electrodes. Impedance spectroscopy measures changes in electrical resistance when target molecules bind to the electrode surface. The amperometric current I is given by NFAD times the partial derivative of C with respect to x, where N is the number of electrons transferred, F is the Faraday constant, A is electrode area, D is the diffusion coefficient, and C is concentration. Now let's discuss why lab-on-a-chip technology is so advantageous for biosensing applications. What makes it superior to conventional methods? First, drastically reduced sample and reagent volumes. We're talking about microliters instead of milliliters, which means less invasive sample collection and lower costs. Second, much faster analysis times. Results can be obtained in minutes instead of hours or days because of the short diffusion distances and efficient heat transfer at the microscale. Third, portability. These devices can be taken to the patient rather than bringing the patient to a centralized laboratory, enabling point-of-care diagnostics in remote areas or emergency situations. Fourth, multiplexing capability. We can design chips that detect multiple analytes simultaneously from a single sample, providing comprehensive diagnostic information. Fifth, automation. The integrated nature of these devices minimizes manual handling, reducing human error and labor costs. Now let me pose a question to you. Why is reducing sample volume so important in clinical diagnostics? The answer is particularly relevant to pediatric and neonatal care. Newborns and infants have limited blood volume, so drawing large samples can be dangerous. Lab-on-a-chip technology enables comprehensive testing from just a few drops of blood. Let's examine a highly relevant case study, the development and deployment of lab-on-a-chip devices for COVID-19 rapid testing. In COVID-19 rapid tests based on lab-on-a-chip technology, a nasal swab sample is collected from the patient. The sample is then placed on the chip, which uses techniques like reverse transcription, polymerase chain reaction, or antigen detection to identify the presence of the virus. Results are available in 15 to 30 minutes, enabling rapid isolation and treatment decisions. Now let's discuss some of the key design considerations and challenges in developing lab-on-a-chip biosensors. Surface chemistry is critical. We need to prevent nonspecific binding of unwanted molecules to the chip surfaces, which can cause false positive results. This requires careful selection and functionalization of materials. Sensitivity is another major challenge. Many disease biomarkers are present at extremely low concentrations, sometimes just a few molecules per microliter. Detection systems must be sensitive enough to capture these rare events. Specificity is equally important. The biosensor must be able to distinguish the target molecule from other structurally similar molecules present in complex biological samples. Sample complexity presents a significant challenge. Biological fluids like blood, urine, or saliva contain thousands of different molecules, and we need to detect our specific target among this complex mixture. Integration of all necessary steps onto a single device requires sophisticated engineering to ensure that sample preparation, reaction, and detection all work together seamlessly. Let me ask you another important question. What is the limit of detection, and why does it matter? The limit of detection, or LOD, is defined as the lowest concentration of an analyte that can be reliably distinguished from a blank sample. It's typically defined as 3 times the standard deviation of the blank signal. Mathematically, the limit of detection equals 3 times signal blank divided by m, where signal blank is the standard deviation of the blank signal, and m is the slope of the calibration curve. The limit of detection determines whether we can detect early-stage diseases when biomarker concentrations are still very low. This is crucial for improving patient outcomes through early intervention. Now let's discuss the materials used to fabricate lab-on-a-chip devices. Material selection is crucial for device performance. Silicon and glass were the traditional materials, borrowed from the microelectronics industry. They offer excellent optical properties and well-established fabrication techniques. Polymers like polydimethylsiloxane, or PDMS, polymethyl methacrylate, or PMMA, and cyclic olefin copolymer have become increasingly popular due to their low cost, ease of fabrication, and biocompatibility. Paper-based microfluidic devices represent the cutting edge in ultra-low-cost, disposable biosensors. These can cost mere cents to produce and are ideal for resource-limited settings. Here's a comparison of the three main material classes. Each has its own advantages and disadvantages depending on the application requirements. How are these intricate microstructures actually fabricated? Let's explore the main fabrication techniques. Photolithography, borrowed from semiconductor manufacturing, uses light to transfer patterns from a mask onto a photoresist-coated substrate. This enables extremely fine feature resolution down to the micrometer scale. Soft lithography involves creating a master mold and then casting polymer materials like PDMS against it. This is much simpler and cheaper than photolithography. Three-dimensional printing enables rapid prototyping of complex three-dimensional channel geometries that would be difficult or impossible to create with traditional planar lithography. Laser ablation uses focused laser beams to directly write channels into polymer or glass substrates, offering great flexibility in design without requiring masks. Now let's discuss one of the most important aspects, clinical translation. How do lab-on-a-chip devices move from research labs to actual clinical use? First, regulatory approval is essential. In the United States, devices must receive FDA clearance through the 510(k) process or full approval through the pre-market approval process, depending on the risk classification. Clinical validation requires extensive testing on large numbers of patient samples to demonstrate that the device performs accurately and reliably in real-world conditions. Manufacturing scale-up is a major challenge. Techniques that work well for making a few prototypes in the lab must be adapted for high-volume, reproducible production. Cost-effectiveness analysis is crucial. The new device must provide sufficient clinical value to justify its costs compared to existing diagnostic methods. User training and clear protocols are necessary to ensure that healthcare workers can operate the devices correctly and interpret results accurately. Let's look toward the future. What exciting developments are on the horizon for lab-on-a-chip biosensing? Organ-on-a-chip devices are creating miniature, functional models of human organs like the heart, lung, or liver. These enable more accurate drug testing and toxicity studies than traditional cell cultures. Wearable biosensors integrated with lab-on-a-chip technology could provide continuous, real-time monitoring of health parameters like glucose, lactate, or stress hormones in sweat or interstitial fluid. Integration with artificial intelligence and machine learning algorithms will enable automated result interpretation, pattern recognition, and even predictive diagnostics. Liquid biopsy platforms using lab-on-a-chip technology aim to detect cancer non-invasively by analyzing circulating tumor cells, cell-free DNA, and exosomes in blood samples. Single-cell analysis capabilities will allow us to understand cellular heterogeneity in tumors and other tissues, leading to more personalized treatment strategies. Here's a visual overview of these emerging technologies. Each represents a frontier in lab-on-a-chip development that could revolutionize healthcare in the coming decades. Let me leave you with one final thought-provoking question. What are the ethical considerations of widespread biosensing? Data privacy is a major concern. Who owns the health data generated by these devices? How is it stored and protected? Could it be used for discrimination by employers or insurers? Access equity is another critical issue. Will lab-on-a-chip diagnostics be available to everyone, or will they increase health disparities between wealthy and poor populations? The psychological impact of constant health monitoring must be considered. Could continuous biosensing lead to increased health anxiety or over-medicalization of normal biological variation? Let me summarize the key points we've covered in this lecture. Lab-on-a-chip technology integrates multiple laboratory functions onto a single microchip, miniaturizing complex analytical processes. Applications span a wide range, including disease diagnostics, pathogen detection, circulating tumor cell analysis, and drug screening. Various detection methods are employed, including optical techniques like fluorescence, electrochemical methods like amperometry, and emerging magnetic sensing approaches. Key advantages include dramatically reduced sample and reagent volumes, faster analysis times, portability for point-of-care use, and multiplexing capabilities. Major challenges remain in achieving sufficient sensitivity and specificity, managing sample complexity, and successfully translating devices from research labs to clinical practice. Future directions including organ-on-a-chip, wearable sensors, AI integration, and single-cell analysis promise to enable truly personalized, continuous health monitoring. Thank you for your attention. Lab-on-a-chip technology represents one of the most promising frontiers in biomedical engineering, with the potential to transform healthcare delivery and improve patient outcomes worldwide.